carbon dioxide vents that I'm showing in this photo here, and the way we ocean acidification system. So, as a community at a local scale and affect individual organisms, scale up to the patterns that we can see in nature. And I think this is especially important in ongoing environmental changes, which organisms, like this common sea urchin, this is Arbachia lixula, which is found in the Mediterranean, have really profound effects on the entire ecosystem, and I'm showing that in the photo. And you can break down environmental change into kind of two broad categories, the proximate effects, and these are what we typically have a better handle on. So these are changes in physiology or morphology or behavior of organisms. But the accumulation of these effects is what we term these emergent effects. So these are changes in distribution or productivity and biodiversity. It's a little bit harder to get at. However, it's often what's the most relevant for society and policymakers when they're thinking about what do we do about environmental change. So understanding these effects has been the kind of central to my dissertation research and what I'm doing as a postdoctoral researcher as well. And I'm going to talk today about the right-hand side of the equation. So this is a rocky reef in the Mediterranean. This is actually taken at my field site by a National Geographic photographer. I am not that good at photography, who let me use it, where we looked at these carbon dioxide vents to try and get at how one of the most pervasive changes in the oceans, ocean acidification, is likely to affect ecosystems. So we use this system together. I worked with Theo McKelly at Stanford, and then my primary collaborator in Italy is this woman, Maria Cristina Gambi, to look at three main themes. So what are the emergent effects on communities? What are the processes that are underlying these effects? And then what do we think the consequences are for the ecosystem or the functioning of this ecosystem? And this is really going to be the outline for my talk. I'll kind of go through some case examples of each of these themes. But I'm going to start with a really brief background on ocean acidification. I know you all are marine scientists, so I won't spend a lot of time here. But to make sure everyone's on the same page, ocean acidification is driven by this increased atmospheric, which is absorbed into the ocean. It reacts with water to form carbonic acid. Carbonic acid is not very stable, so it readily dissociates into hydrogen ions and bicarbonate ion. This increase in the hydrogen ion, that's where ocean acidification gets its name as the water becomes more acidic. But that is not the only important part of ocean acidification. So another thing that we have a lot in is the formation and dissolution of calcium carbonate, which is dependent on carbonate ions and calcium ions. So unfortunately, a lot of those hydrogen, or fortunately, depending on which organism you are, a lot of those hydrogen ions actually react with the carbonate ions, and you get more bicarbonate. So the end result is threefold. You get this increase in the hydrogen ion and the decrease in pH, an increase in the bicarbonate ion concentration, and a decrease in the carbonate ion. So these changes can have a variety of effects on a wide diversity of organisms. And I'm just kind of highlighting the really key things here. So the changes in the acidity can affect the acid-base balance of different organisms. Some organisms can cope with this, but that is likely to have effects on their energy budget and their metabolism. A lot of the marine plants and algae use bicarbonate or carbon dioxide, so the increase in that bicarbonate ion concentration can actually benefit the photosynthesizers. And then last, what's really garnered the most attention is the carbonate ion concentration. So decreases in this can affect decrease calcification or increase the dissolution of calcium carbonate. And this is really what's garnered the most attention because so many organisms in the ocean use calcium carbonate. So they range from the coccolithophores, which are really important for biogeochemical cycling, to pteropods, the base of food webs, to commercially and ecologically important species that you all know and care about. However, the effects are much broader than what we think about on calcifying organisms. So this is the results on that meta-analysis that Bill mentioned, where we, together with some collaborators from UBC, we collected all the papers that we could find in 2010 that manipulated seawater pH or carbonate chemistry and measured some kind of biological response. And I'm just showing you the response on growth. So each one of the data points represents the response to a 0.5 unit decrease in pH, so kind of what we expect in the year 2100. And the way you can interpret this is that a negative response variable means a reduction in growth. Positive is the opposite, and significance is determined by those confidence intervals. I also have some little parentheses in there. That's how many studies at the time had gone into calculating that mean effect size. So the really broad pattern is that indeed calcifying organisms do appear to be more sensitive than some of the non-calcifying organisms. However, we do see in general, some of the marine plants or algae are benefiting from acidification and actually grow much faster. And there is some variability even within the calcareous species. So here you see some of the really highly mobile active species tend to do better. And we think this is because they have a lot of the cellular machinery in place to deal with the buildup of CO2. That's common when they're active and they're building up CO2s in their cells, so they can also deal with regulating their internal pH with acidification. These are the predictions from the CAR Community Climate System model. This was a few years ago. I'm sure we'll see some new projections in the new IPCC reports. But these are their projections for about a 0.4 unit decrease in ocean pH over the next 85 years. And what I want you to get from this really is just that ocean acidification is predicted to occur across all of the world's ocean. This is really a global stressor, and virtually all marine ecosystems are likely to be impacted by some kind of change in their carbonate chemistry. So given how pervasive the stressor is, and that we see these diverse effects across a really wide range of species, there's a lot of interest right now in how does ocean acidification, how is it likely to affect marine ecosystems? And while most research is happening on this left-hand side, and we are getting a much better understanding about what it means physiologically for organisms, we don't have a great handle necessarily on the right-hand side of the equation. One way to get at this is to use these carbon dioxide vents that I used for my dissertation research. So these are shallow water carbon dioxide vents. They emit about 90 to 95% CO2. There are trace amounts of other gases with them, but there's no sulfur, nor are there any changes in temperature. So a lot of times when I talk about them, people are picturing in their mind these deep sea hydrothermal vents, but they're not. These are in 2 to 3 meters of water. They're in really typical rocky reef communities. It's more like swimming through a glass of champagne or a jacuzzi without the heat on. So you've just got carbon dioxide bubbling out of the substrate. Or if you do ocean acidification research, this is like a gigantic bubbling experiment that you would do in the lab. So these are found in the Mediterranean Sea, in the Bay of Naples, right off this island of Ischia, at the base of this 14th century castle. This has nothing to do with my research, but it's kind of fun to look at. This is where I got to work, which is fun. And there are gradients of the vent activity on both the north and the south side of this island. And by gradients of vent activity, I mean amount of bubbling. So we have areas with a lot of bubbling that have extremely low pH, moving into areas with no bubbling over about 150 meters. So each of these pH zones that I'm showing here, those are about 50 meters long. So I talk about these as pH zones, and I will throughout the rest of the talk, but really these are driven by an increase in the dissolved inorganic carbon, and you've got pretty constant total alkalinity and salinity. And the reason I say this is just that it means that the changes in all of the carbonate parameters are very similar to what we expect with ocean acidification that's driven by increased dissolved inorganic carbons. So, and I can talk to you more about what those values look like, but I do end up referring to them as pH zones. So these are some of the values from, these are using in situ pH sensors out of the Scripps lab of Todd Martz. And what you can see is that in the ambient pH zone, we see pH values that are very similar to the surface conditions in the Mediterranean today, so around 8.0, 8.1. There is a little influence from the vents as water sometimes washes through there. In the low pH zone, they're closer to 7.5 to 7.8. This is what's most comparable to what we predict for the year 2100. And then we have these extreme low zones, and these are very acidic zones, so it's really beyond our predictions for the near future, but they give us a good kind of end member scenario of what acidification is doing to communities. Okay, so switching gears, I'm going to talk about this first study where we just use the system to look at what are the emergent effects on ecosystems when we start thinking about complex assemblages with interacting species. Okay, so these vents are found in really typical rocky reef ecosystems, much like the kelp forest around here, algal dominated, but we often refer to them as miniaturized systems. So the canopy in a rocky reef system in the Mediterranean is about yay high. And they're really highly diverse, but the invertebrates within these communities are about that big. So it's a really miniaturized system. Think about your kelp forest and shrinking it way down. And the focus of this first study was on a lot of invertebrates. So these are the things like the gastropods, amphipods, polychineids, the little guys that are in that canopy. So what I was interested in this first study was just how does the variability that we see in species responses in labs, how does that play out when they're in a complex community and they're interacting with one another? Do we see the same patterns, or is there divergence from these patterns that we see in the laboratory? So here I'm showing you kind of the key groups that we worked with at the vents, starting from the heavily calcified species on the left, so I've got gastropods and bivalves, to those less calcified species, the crustaceans, and then most of our polychaetes were not calcified, although there are a few species that are. And on the y-axis, I've got how we think they will respond, or their abundance will respond to acidification. So based on a null hypothesis, or what we're seeing in the meta-analysis, these laboratory studies, we can make some predictions that generally we think those really heavily calcified species will do worse in acidified environments, and then some of the crustaceans, those highly mobile ones, will do okay, or we won't see any trend with them. An alternate hypothesis is that if species interactions are important in this community, that we might see compensation. So as the calcified species drop out of the system, some of these more tolerant species come in and take their place. So to do this, it's a pretty simple study. We just went out and collected all of the benthos in these 20 by 20 centimeter plots in each of the pH zones at each of the sites. To do this, we used a glorified underwater vacuum. It's called an airlift. Maybe some of you guys use it here, where more or less you hook up this tube to your scuba tank, and it sucks all of them into a little mesh bag so they can't swim away. Then we collect all of the benthos, we scrape it off, and look at everything under a dissecting microscope to try and find all of those tiny, tiny little amphipods and count them and classify them to species, which is no small task. Okay, so here are the results. There's a lot going on here. One of the first things I say is that I'm going to use these same colors throughout the talk. So red, extreme low, bad, we hope we never get there. Yellow, danger, we're moving into acidified water. Blue, that's ambient, that's where we're at right now. So the first thing that you can see is that some of these heavily calcified species like the gastropods do drop out in the extreme low pH zone. In contrast, some of them do okay. Some of the species that we think would do well drop out. And then there are some species that show their highest abundances in these really extremely low pH conditions. So to simplify this a little bit further, we see a couple species that do drop out in extreme low pH zone, but then we see two species, or I shouldn't say species, these are taxonomic groups. I'm grouping them back up here for you guys. We see some of the taxa that show their highest abundances, and then some with no trend whatsoever. So what this suggests is the kind of variability that we're seeing in lab experiments is likely to be more pronounced in multi-species assemblages when they start interacting with each other. So we are likely to see compensation with some of the more tolerant species, and then some of them that we think may be resilient might not actually do so well when they're in an environment and reliant on other species or food sources that are impacted by acidification. Mechanisms underlying this are complex. This is a species, I think I had 125 species in this community, so we're still working on trying to figure out what is actually driving some of these to increase in abundance and decrease. I think the high variability is something that we should expect. So the second thing we want to know is that some of these taxa that are increasing in abundance, can they actually stabilize some of the community parameters? So now I'm grouping all of them together. These are the number of individual invertebrates across the 3 pH zones. And you can see this evidence for the density compensation. So there are the same number of invertebrates in these really extreme conditions as there are in the ambient conditions. However, there's much less biomass, which is what I'm showing on the right-hand side. So you can break this down again by thinking about which groups. So I'm showing the same exact graph. Now I've got the abundance or the number of individuals on the y-axis. But I'm highlighting kind of three main groups, the mollusks, crustaceans, and polychaetes. And what you can see is that, as we saw in those previous graphs, the species or taxa that are compensating or able to increase their abundance in the extreme low zone are primarily the crustaceans. But those crustaceans are super small-bodied, opportunistic species that make up very little of the biomass. So even though they're able to cope in these conditions, it's a very different community. We have much lower invertebrate biomass. And many of the fish species that we eat and people eat in the Mediterranean and base their livelihoods on are eating invertebrate species. So it's a community reorganization to an entirely different community that's likely to have different services. And in general, we see a reduction in diversity. We don't see any new species in these extreme low pH zones. We're just losing species as we move into these more acidic environments. So for this section of the talk, really what we found is that indeed some of these really heavily calcified species are very sensitive to acidification, as we predict. However, species interactions do appear to enhance variability, and we should expect compensatory dynamics. The end result are really simplified assemblages in acidified conditions with reduced diversity and reduced biomass, which is likely to have implications for energy transfer and higher trophic levels. Okay, so this is just one example of an acidified ecosystem, and we don't have very many. There's another one in Papua New Guinea, and people are trying to kind of find another one in the Mediterranean. But in order to start thinking beyond this community, really we're interested in what are the processes that are underlying these effects or the community reorganization. So for this section, I'm going to talk about the sessile benthos. So in the patterns that I just explained in the mobile invertebrates are in the sessile benthos. So these are two pictures taken at the site in both the ambient and acidified conditions. And in general, we see a reduction in diversity. We see an increased dominance by species. In this case, it's the fleshy algae, some of the turf-forming and erect brown algae, and reduced abundance of some of the calcareous species like crustose coral algae. And there's been this kind of broad assumption when we started characterizing the patterns that the reason we don't see calcareous species in the acidified conditions is because of the direct effects on their ability to calcify and grow. However, we wanted to test the alternate hypothesis is what is the role of species interaction in actually driving or affecting the abundance of calcareous species. So one way to get at this is to look at succession. So I'm showing here a picture of, well, if I can get us back there. So this is a tile in the ambient pH zone that is growing, where we're growing a community over about the period of a year. And by watching how the community forms through time, we can get some insight into the importance of these ecological factors like recruitment or species interactions in actually organizing or developing the community. So again, pretty simple methods. What we did was put out these 20 by 20 centimeter tiles in each of the pH zones at each of the sites. And these are them in the low pH zone with one of the pH sensors we used for the carbonate chemistry. And then we collected a subset of the tiles every few months for the period of about 1 year, or 14 months. Then took them into the lab and we tried to classify down to species where we could, but I'm grouping back up here again for the analyses, into these broad functional groups. So these are things like the crustose corallin, biofilm filamentous algae, erect brown, grazers, filter feeders, and so on. We also measured the relative growth rates of some of our key calcareous species. So the two main groups here are the crustose corallins and the barnacle species, Balanus perforatus is our main species. And for these guys, what we did was identify individuals or discrete patches of crustose corallin algae at the early stages of succession, and then we used photoanalysis software to follow those individuals or patches through time and estimate their growth. So I'm going to jump to the end. So this is after 14 months of development, so about a year. We do see that the community on the tiles differ between the pH zones, and they're very reflective of what we see in the background community. So in general, we have more calcified taxa and fleshy algae in this ambient pH zone. In the low pH zone, we see increased dominance by the fleshy and turf algae, and in fact, there's a tile under there that's pretty hard to see. And then in the extreme low zone, these are really depauperate communities that are dominated by biofilm and filamentous algae. And in addition to just the resulting community at the end of the time series being different, the patterns or the successional trajectories to these communities were really different among the pH zones. So this is an NMDS plot. Each of the points represents a community, and then distance on an NMDS plot represents differences in those communities. So I've put time 0 down here on the bottom left. This is the blank tiles, and then I've connected each of the time points with the black line. And this is just a this is the mean community structure in the north on the extreme low. And what you can see is that there was this really big colonization event. So we get a big difference from a blank tile to colonization, but then there's very little development through the rest of the entire year. So pretty much the community established every 1.5 months, and it never really changed. This is different than what we see in the low and the ambient pH zones. So here, we see the same initial colonization, and in fact, it's very similar to what we see in those extreme low pH zones throughout all of succession, but then there's continued development. So then you see the community continue to change through time. And in fact, in the ambient and low pH zones, they're very similar until about 6 months when they start to diverge and look different. So we can get some insight into what's happening in succession by looking at these functional groups. So now I've got the percent coverage of filamentous algae on the y-axis, time on the x-axis, and I'm using those same colors. And you can see that all 3 zones show this really high percent coverage of biofilm filamentous algae during the early stages. These are gradually replaced by other species in the ambient low pH zone, but they persist in this extreme low pH zone. And this really just suggests that in these really acidified environments, they're just stuck in this early successional stage with rapid growing, fast colonizing algal species. So exploring things a little bit further, now I'm showing Calcareous Taxa on the y-axis. So I'm grouping together those crustoscorallins, barnacles, spirorbid polychaetes, time on the x-axis. And a couple things are clear here. One, we never saw, ever, calcareous species in the extreme low pH zone. So this would suggest that there are instances when the conditions are so severe that it's probably physiological intolerance or lack of recruitment, so that we never really see calcareous species. They can't survive in these conditions. However, in these low pH zones, which is what is really interesting if we're thinking about near future predictions for the year 2100, we see actually very similar recruitment and very similar growth rates for the first 3.5 months in the low and the ambient pH zones. And it's not until about 6 months that we see this divergence, where we see higher abundance of calcareous species in the ambient pH conditions. And this is also clear in the growth rates of the calcareous species. So this is for about the first 3.5 months of succession. I've got the relative growth rates of crustoscorallin on the left and the relative growth rates of barnacles on the right. And we don't detect any differences in their growth rates in the ambient low pH zones for the crustoscorallins, and a really small one for these barnacles. And this does not make up the difference between the amount of crustoscorallins on the ambient and low pH tiles. So this suggests is that the decreased abundance of calcareous species in these low pH zones is probably not driven by these direct effects on calcareous species or their ability to grow, or at least that's not them. So we can get a little bit more insight by comparing it to some of these other functional groups. So now I'm showing you that it's the same graph as I was showing you before. I've got calcified taxa up here. I've now just expanded this out a little bit more so you can compare it to the turf algae, which is fleshy turf algae below. And what you can see is that when we see the ambient and low pH communities differ in their calcareous species is at the same time when we have this really rapid overgrowth by fleshy turf algae. So when I would go out and visit my tiles for the first 3.5 months, you couldn't tell any difference between them. If I looked at a tile from one zone to the other zone, they looked very similar. But at about four and a half months, actually, we had fleshy turf algae come in and colonize, and they just overgrew everything in those low pH zones till they were like the photo that I was showing you earlier, where you couldn't even find the tiles in those pH zones. What this suggests is that we don't have strong evidence that the decreased abundance of these calcareous taxa are driven by direct effects, that they can't stand these environmental conditions, but instead that their abundance is likely impacted by interactions with fleshy turf algae, which may be benefiting from these same conditions, likely due to increases in the bicarbonate or carbon dioxide that they can use for photosynthesis and growth. So together, all of these results suggest that at least in extreme acidification scenarios, recruitment and physiological intolerance can limit the abundance of calcareous species. However, it appears that altered competitive dynamics may be a really critical factor in driving community patterns in these conditions that we're predicting for the near future. The end result is different competitive dominance. So in the extreme low pH zone, you have these fast-growing, opportunistic filamentous algae, which is in contrast to the low pH zone where the calcareous species are outcompeted or overgrown by the turf algae. Okay. So ultimately, I'm interested in what does this actually mean for the ecosystem? How does this ecosystem function differently? So I'm going to change gears for a minute, and I'm going to talk about a study where we looked at some of these larger patterns to try and get landscape scale diversity. So Mediterranean rocky reefs have high natural variability in both space and time, and we refer to this as habitat patchiness. And we know in the Mediterranean there's been a lot of research that habitat patchiness is driven primarily by grazing or sea urchin abundance. So urchins come in, they graze down those fleshy turf algae, they create space for either competitively inferior species to colonize and persist, or open space for those crustose coralline algae to thrive. So given that sea urchins are calcareous and we're worried about how they might deal with acidification and the patterns that we're seeing at this site where there's this increased dominance of the fleshy turf algae, we wanted to know how does acidification affect the habitat patchiness of the ecosystem. So for this, we looked at the recovery from disturbance. So what we did is we cleared these 20 by 20 centimeter plots, and then we watched how the community developed or recovered through time. And this allowed us to characterize the variability between these plots, starting from the same initial conditions of bare rock. So in particular, what we did is we cleaned these off with wire brushes. We tried to get everything off of the plots and each pH zone in the sites, and then we photographed them through time for about 3 years. In addition, we monitored the sea urchin populations along permanent transects. There are two common sea urchin species here, Paracentrotus lividus and Arbachia lixiula that I showed you that photo of earlier. And we counted them and measured them for about 3 years along these same transects. So our hypotheses based on what we know about Mediterranean rocky reefs. Under ambient conditions, we expect to see a lot of urchin grazing. We expect to see episodic disturbances and high habitat patchiness or high variability in the assemblages. If acidification affects sea urchins or their grazing or their behavior, then we would expect to see reduced grazing, lower disturbance rates, and just more homogeneity in the assemblages. So again, I'm going to jump to the very end. This is after about 3 years. And what I'm showing you here are individual plots in each of the pH zones. So each bar represents a single plot. And then the composition of the community in that plot is represented by the different colors. So for example, fleshy turf algae is shown in green. And the first thing I want you to see is that we have a lot of variability in the ambient pH zone. So there are a lot of colors here. Each of the plots was a little bit different from one another. This is in contrast to the low pH zones where we saw this really heavy dominance by the fleshy turf algae like we saw in the tiles. then I know you guys are probably going to ask me about this, so I'll just tell you. We do see a little bit of patchiness in this extreme low pH zones. There's two types of communities. We've got the biofilm filamentous algae, and then those that have fleshy turf and erect brown algae. And this is kind of a habitat patchiness, but it's very different from what we see in the ambient pH community because these are two really simple depauperate communities versus a range of different developed communities in the ambient pH zone. It's persisted throughout the 3 years where we see this higher variability in the ambient pH zone, which suggests that indeed we do see more habitat patchiness or higher variability in ambient conditions. So again, we can look at these groups. Again, showing similar colors. Now I've got biofilm filamentous on the y-axis, and like we see in the tiles, there's a lot of colonization by these species across the pH zones. Again, they're replaced, but they persist in this extreme low pH zone. In contrast, we see something different in the low pH zone. So again, there's a little bit of noise during these early stages of succession here, but we see this kind of canalized recovery where the community is converging on this really high abundance by turf algae through succession. So this suggests that when we're looking at both the benthos and the tiles, that this may be a fairly robust pattern that we can expect to see with this increased competitive dominance of the fleshy turf algae in acidified conditions. Okay, and then the last thing I'm going to show are the percent cover of the crustose corallines. And this is really just to highlight the kind of variability that we see in these ambient pH zones. So we see these sharp spikes, and this represents the really high variability through time where you have low percent cover, and then all of a sudden there's a lot of percent cover. And there's a lot of variability through space as well, and that's these really large error bars associated with the ambient pH zones. And as we expected, really high variability was associated with sea urchins. So in the ambient pH zone, I would swim up to my plots, and the plot that was 2 months ago covered with fleshy turf algae would be right next to a little urchin. This is the plot here. I have these two little blue corners. And all of the fleshy turf algae would be gone, and we'd have these clearings, and new species would be coming in. In contrast, we never saw them in the low pH zones. We saw a lot of urchins, but we never saw this really clear grazing sign. But what's really interesting, I think this is an interesting thing, is that this is the abundance of both species, so Arbachi on the left and Paracentrotus on the right. We never see them in extreme low pH zone. They just are never there. But we see very similar abundances across the pH zones, both the ambient and the low pH zones, in both species. So neither of these differences are significant. Similarly, we see really similar sizes of them. So they are big, healthy-looking sea urchins in these low pH zones. And this is really contrary to what people would expect, because we have a lot of research that say, well, sea urchins appear to be pretty sensitive, and they're calcareous, and they use a really sensitive form of calcium carbonate. Despite the fact that we see large abundance of sea urchins in the low pH zone, and we see similar sizes, we don't see evidence of sea urchin grazing in these pH zones. So this is the amount of halo. And the way we defined halo was an area of clearing that was typically associated with was always associated with a sea urchin. They're typically kind of near a crack where the sea urchin likes to hang out. So they're just going out there, they graze the area, and they go back and hide in their little crack. So we see a lot of this grazing area in the ambient pH zone, but a lot less of it in the low pH zone. So this would suggest that the urchins are there, they're growing, but they're playing a different functional role in the low pH zone. So they're not creating the same grazing areas, halos, opening up free space for habitat patchiness and maintaining diversity. I should say that the areas that we're moving forward is trying to figure out, well, what are the mechanisms that are driving this? So this could be that urchins are grazing less, could be that fleshy turf algae are growing faster, that the urchins are still grazing, but that turf algae is just coming back in and covering up any signs of their grazing, or a combination of both. And that's a direction that we're trying to go and figure out what is happening there. Okay, regardless of the mechanisms, this study showed us that we do see or disturbance regimes. There's less signs of urchin grazing in these low pH zones. This results in less variability in the community, so more homogeneous assemblages, and reduced diversity at this larger landscape scale. So in terms of at the beginning of trying to understand what are emergent effects of environmental change, or in this talk of ocean acidification on marine ecosystems, species interactions, of considering species interactions when we look at larger scales. One, we're seeing that species interactions are likely to cause a community reorganization. So it's not that we're just going to lose calcareous species and they're going to drop out, but we should expect to see these compensatory dynamics or increased sensitivity in species that we may not expect because they look to be like they're doing fine physiologically, but they drop out because something that they rely on also drops out. And one of the things that I think is really important is that altered competition could represent a, we're talking about fleshy turf in their lab experiments about reduced growth of calcareous species or increased growth of fleshy turf algae. When you put those things together, those small scale changes can shift the competitive balance such that you get these really large scale changes that if any of you went and snorkeled over the site, it looks really different in the low pH zone than the pH zone. And then last, our research suggests that the role of calcareous herbivores could be altered under acidified conditions. And this is important because a lot of the really important herbivores in marine ecosystems are calcified, from kelp forests to coral reefs. And if acidification changes their abundance patterns or their behavior or their ability to graze, this could have implications for other ecosystems as well. And it is an area of important research. The last thing I want to leave you guys with was just a visual from these vent sites, because I think they provide a really important stark visual of what it means to be an acidified ecosystem, where we see just a general reduction in diversity, simplified ecosystems. This is what a simplified ecosystem looks like when we talk about it. That's likely to have implications on some of these higher trophic levels, so reduced invertebrate biomass. And I think it's just a useful tool for all of us to think about what kind of ecosystem changes should we be thinking about in the future with environmental change. So lots of people helped with research. I'd like to thank them, and I think I have time for questions. Yeah. Yeah.
I have two questions.
Yeah.
First, I'm wondering if you think maybe the urchins are not actually being affected so much by their ability to handle the change in pH, making them eat less or move around less, but what if it's just that the algae are growing so fast they don't have to go anywhere to chop down?
Yeah.
Eating fine.
Yeah.
And my second question is just kind of a left field-y kind of question is if this is where we're heading, do you think people need to start thinking about developing herbicides for underwater applications to try to manage this kind of system?
So I haven't thought much about the second question. The first question I've thought a lot about. Yes, I think they could be doing selective grazing, where, especially like the urchins here, they can actually be eating drift algae or things like that. We don't see as much drift algae in this system, but I think they could just be grazing differently. So they might be creating smaller halos, and the turf algae is just coming back, or they don't need to go very far to get enough. The other hypothesis that I have is just that they have a reduced metabolism, so they don't need to eat as much. But we don't have very high, you would expect to see a change in the size spectrum. We don't see that. And my thought about that is that there's not a lot of predators in this system. So the Mediterranean, those of you who have been there, people have been fishing this system for thousands and thousands of years, and there are very few big fish. So a lot of the predators for sea urchins are gone. So potentially that there's not a big cost for them growing slower. They're just not eating as much and kind of hanging out. What's interesting is that these urchins in the low pH zone, you know, I'd like move some in there and kind of watch what they do. And more or less from year to year, you can find them the same. They're just like hunkered down, not doing much at all. So we're trying to figure it out. In terms of herbicides, I thought more about, well, ocean acidification is this really large-scale problem, and it's really hard for individuals to do much about it. But we do know the other factors that promote algal overgrowth, so loss of herbivore populations or increased nutrient supply. So my focus has been more on if we deal with some of those things, we're less likely to exacerbate the effects of acidification. So make sure we have healthy herbivore populations, you know, figure out your fishing regulations and try and deal with other things that push us towards algal-dominated ecosystems. Hi.
That's really great, thanks. I'm interested, you talked a lot about the urchin herbivores. And since you're seeing this increase in fleshy algae, you didn't mention what was going on with other molluscan grazers and
Yeah.
what's going on with them. And did you see them, and did you look for them at night? And because there could be some other changes in their grazing behavior.
We didn't get at grazing behavior with any of the grazers. So we have a proposal out that we want to try and do that. The other mollusks and grazers, we can't detect any differences between the ambient and low pH zones. They're there. And I think that this is an interesting thing to think about, is that a lot of our research shows these reductions in growth and calcification of these heavily calcified species, but most of them are there in the low pH zones, and this is kind of our predictions for the year 2100, but I think that system is very close to moving over into this really depauperate community. So we see abundances, limpets are there, gastropods are there, but they don't, I think on the tiles, we never urchins get on the tiles. So I think the grazers on the tiles are primarily the limpets and the little mollusks and things like that. So it could be, again, that they're not grazing in the same way, but we haven't figured it out. Yeah, I'll go Walton and Scott. Yeah.
That brings up an interesting question. So your low pH zone is your prediction for 2100.
Yeah.
It's actually not too far away.
Yeah.
And then you have your extreme low as the extreme. But in time, you might consider it being linear. But from an ecological perspective, do you think there's maybe a tipping point where it's much closer to get to that extreme than a linear progression of time?
Yeah. So this is, it brings up some of the logistical issues with this site, is that the site itself is not a really good gradient. It really is more zones, because you have areas where there's a lot of venting and then little and then none. So we're proposing to do some research where we're doing really high resolution carbonate chemistry along the coast, so we can try to get at what's the functional relationship. Is there linear changes? Are there tipping points? My sense is that there are some tipping points. I mean, it is so distinct. It's really interesting when you're swimming along. It's just like, wow, we've moved into a new kind of community, and it's like a line there. And that doesn't totally correspond with all of a sudden heavy venting. It's in the area. So my guess is that these are nonlinear dynamics, that there are likely tipping points. But you know, the other interesting thing is that when I talk to climate scientists, they don't like the extreme low pH zone. They're like, that's so extreme, we're not going to get there. So I was like, yes, it's an extreme scenario. It helps us understand the system. But when you talk to paleoceanographers, a lot of paleoceanographers, and I'm sure there's some of you out there, have said, well, actually, that's kind of what some of these systems that I study look like fleshy, algae-dominated, depauperate, no calcareous species. I don't know how far off that is. So. Yeah.
So I was just curious what you thought would happen if you started with communities that were characteristic, like a fully developed ambient community, and then put them, you know, in the different zones.
Yeah.
You know, would the calcareous species potentially be more tolerant if they're already established, or you know, what kind of dynamics do you think would happen in that case?
Well, I can tell you we did that with urchins. They seem to survive just fine, but they, like I said, they just hunker down and sit there. In terms of the whole community, it's hard to say. what would happen? I think you would see slightly different things, because when the fleshy turf algae comes in, they're not fighting against very much. They've got these bare substrates where there's, you know, some crustose coralline and barnacles and things, so it's really easy for them to overgrow. I don't know that they're going to outcompete a fully developed community. But if we think about it in terms of physical disturbances are really common in these systems, and we have patches happening all the time because of storm disturbance or whatever, then that's where I think you're more likely to see these fleshy turf algae come in and start to dominate. Whether they can outcompete like an articulated coralline algae that's big and happy, I'm not so sure. I'm not sure about that. Yeah.
Just in how important variability is in the system.
Yeah.
I don't know much about the med, but from what you described, so it sounds like those pH zones are pretty stable, which is very different from what you see in an upwelling system up there.
Well, then.
The upwelling and
Yeah.
really dramatic changes in pH over a very short temporal spatial scale.
Well, then I might not have done a great job explaining my pH zones because they are stable, but there's a huge amount of temporal variability. I might have a photo of this. So here you go. Okay, so that's the pH through time for about a month. So those are mean values that I'm showing you, and there's the variability increases as you go from the ambient to the low to the extreme low, and it's very, we've done a whole bunch of time series analyses on this. There's not big tides in the system. This is just like those low pH waters are getting washed around and in there, and then they sit there for a while. So this is actually much more variable than most systems. That's a question of interest to me, is that are we underestimating effects because there are these sharp fluctuations, and so there's like a physiological release or rescue when it comes up and they can just hang out for a little while and then it gets bad? Are these things being driven by these really low minimum values? Is mean really the best way to go about it? Those are definitely open questions. That's another thing that we're trying to do with this really high resolution monitoring of the system, is to see if we can start using some statistical techniques to pull out, is it a mean? What describes the pattern? Likely it's probably all of the above. But these are probably more variable than even an upwelling system. Yeah, because it's so rapid. Yeah.
Sorry, I missed the beginning.
Yeah,
Great talk.
thank you.
With respect to Erica's hypothesis that these algae may be just growing more quickly, and the one hypothesis would be that there's just more carbon available, so their carbonate concentration system is limited. It seems like there'd be a really strong isotopic signature to that gas phase versus the inorganic phase. Has anyone looked at del-C13 signature of those?
We did a little bit.
Okay, because I think that would tell you whether these vents are promoting growth or not.
We should talk more. I can show you my initial, I kind of, 8 years ago or something, I did a bunch of isotope stuff and then ended up going in different directions. But it's something I'm interested in, seeing if we can figure out that signal.
Back to the isotope.
Yeah. Yeah. Any more questions?
Do one more, maybe? I had a question.
Yeah.
You talked about top-down, someone else. The idea that higher trophic predators or herbivores may actually drive the system more strongly. And have you thought about trying to do any inclusion experiments here, or could there be any way to kind of bring back those, say, stop fishing or do net inclusions?
Would be hard to do in this system. No, we haven't. I mean, so this system is really hard to get at fish or these higher trophic levels because they're so small. You've got these 50-meter zones, and the fish are just like in and out. I mean, we have fish. We don't have big fish. They're all little. They're miniaturized, but not because it's the Mediterranean naturally, because they've been fished down. But they're going in and out. They don't seem to be bothered by the acidification or by the bubbling at all. But it's so unrealistic because they can just leave the zone whenever they want. You could potentially, it's a really small area and there are fishermen there, and it would get torn to shreds. Pretty much most of your experimental stuff gets ripped off, so you'd have to be there watching it. Yeah. Thank you very much for having me. Thank you.
Cool to see like the next evolution of your talk after
Yeah.
like going to your dissertation.
Yeah.
It's like it's got that next.
It's got another layer
But I
on
wanna
it.
see your paper in there.